Good day. In this lecture, we will be talking about what other entities are present in the microbial world aside from those cellular entities. So this is about viruses and other acellular agents, focusing mostly on the infectious ones. So the most popular and most well-known other acellular entities in the microbial world are the viruses. And the reason is because many of them cause diseases because they are parasitic. So typically thought to be the major cause of disease, but aside from being disease causing, we have found ways in order to use viruses. So viruses are considered vital members of the aquatic ecosystems, and recently they are being used not only to destroy cancer cells but also to develop vaccines against other viruses, and although to perform other biotechnology techniques like recombinant DNA technology. Bacteriophages in the human guts may be known to regulate bacterial microbiome structure because viruses does not only infect us humans and other animals or plants, but they can also infect other cellular organisms like bacteria. So they help keep the population of the bacteria in check. And as such, they are very good places to study. In fact, in the field of biology, we know a lot about viruses because we use them. They have many different uses. They are tools. They are part of the toolbox of biotechnologies and bioengineers. So they are very important organisms. So viruses are a cellular structure. So when we say a cellular, they do not have cells. They are not composed of cells. They are made of a different structures. So virology is the study of viruses, and we define viruses as infectious agents unique in their simple cellular organization and pattern of multiplication. So one thing that separates viruses from basically mundane molecules and objects is that they can multiply. So, and viruses are, of course, known to be major causes of disease. In fact, we have just recently come from the pandemic of 2020. This is a world, uh, the entire world basically have woke up to the reality of the virus. So, hence, they become very important model systems, especially in molecular biology, biotechnology, and bioengineering. So we have, excuse me, extracellular and intracellular viruses. So viruses are not considered living organisms. The reason is because they, outside of the cell, they are considered non-living. They are inactive, they cannot produce, they cannot perform any metabolic activities. They are basically inert. They, we don't call them sleeping. They are basically just objects like, I don't know, just like a computer that is turned off, shut off from its power. However, inside the cell, they become active. They commandeer the whole cell and use them to synthesize their own components, which mature progeny viruses are assembled and released. They use the cell's machinery in order to propagate themselves. And that's all. They just want to propagate themselves inside the cell. So as very small and simple structures, they can infect cell types, all cell types. So for the bacteria, we call the viruses that infect bacteria as bacteriophages, or in short, phages. So aside from the bacteria, they are also known to infect archaea bacteria or archaeal organisms. But again, not much is well known for them. So we know most of the viruses as either phages or the eukaryotic viruses. So for the eukaryotic, they infect plants, animals, protists, and fungi. So here's an example of a phage. This is a T even coli phage, and we have this mini virus. So the virion structure, if you look at it, is defined by their capsid symmetry. This is their outer covering, and they are symmetrical, and they do not have, or rather, it's either a presence or an absence of an envelope. An envelope is a structure surrounding this capsid. Sorry. So you have this, for example, this is the adenovirus with the capsid, and this is an envelope virus You have the capsid inside, then surrounded by an outer envelope. In fact, the SARS-CoV virus is an envelope virus. So the structure of viruses, the mature virus particle, we call them the virions, and we usually observe these virions outside the cell. Inside the cell, they do not form these structures except when they are being or they are set to be released outside the cell, but in its intracellular form, they do not have this form basically. So the virions range in size from 20 nanometers to the size of a rod-shaped bacteria. So some viruses can also be viewed by, or rather, because they are very small, they must be viewed by an electron microscope. We do not really observe them much with a light microscope. So what's inside a virion? The virion contains a nucleocapsid, or we call them a capsid, which is their protein coat. So you have this nucleocapsid, which is contained contained therein is their quote unquote the nucleus, which is their genetic material. Now, the genetic material of the viruses is, or rather, can be DNA, a double-stranded DNA, a single-stranded DNA, or even an RNA, which can be either a double-stranded RNA or a single-stranded RNA. And for the single-stranded RNA, they can be plus sense or minus sense RNAs. So protecting this genetic material is the capsid. So all in all, this structure is the nucleocapsid. And then we have some viruses contain a lipid membrane envelope, just like the SARS-CoV virus. And then we also have the naked virus without a nuclear envelope. So this is just a simple virion. So you have the capsid, and this is their genetic material. And then you have the envelope virus with a lipid bilayer, a lipid membrane surrounding them. So the sizes vary also. We have a herpes virus, the rhabdovirus, and the T fudge or the coli fudge, the event coli fudge. These are quite small. A very small human papillomavirus. You have the sulfolobius spindle-shaped virus and a quite large mini virus. And then we can also have a two-tailed virus. So let's look at the protein coat, which is their capsid. So the capsid is a large macromolecular structure which serves as the protein coat of this virus. They are made of protein subunits called the protomers. So they protect the viral genetic material and aid in its transfer between host cells. So non-enveloped viruses construct a capsid. So a capsid is constructed from many copies of a single protein, and that is actually one of the reasons why they are quite symmetrical. So envelope viruses require nucleocapsid proteins and extra proteins to anchor to the membrane. So just like your SARS-CoV virus, which is an envelope virus, they have spike proteins in their membrane that allows them to anchor themselves and integrate themselves into the cells. So the capsids are helical, icosahedral, or can be complex structures. So the helical capsids are shaped like hollow tubes with protein walls. So the protomers self-assemble into a rigid tube. The size of a capsid is influenced by the promoters and, of course, the genome of this viral genome structure. So here is a rod-shaped virus, which is actually made up of this genetic material, the RNA, um, which is protected by this capsid. The capsid is actually a multi-subunit protein shell enveloping them. So for icosahedral capsids, this is quite an efficient way to enclose a space. So you have an icosahedron shape with 20 equilateral triangular faces and 12 vertices, just like a D&D 20-sided dice, basically. So capsomers is the ring or knob-shaped units made up of 5 or 6 promoters. So these are the subunits of your capsid. So we have pentamers, which are promoter capsomers, and the hexamers for 6. So pentamers and hexamers for 5 promoter capsomers, hexamers for 6 promoter capsomers. So basically they form these icosahedral capsids. So some capsids does not conform to the icosahedral shape. They have quite their own complex symmetry. So some of those examples, we have this pox viruses, which is the largest of these animal viruses, and we have also large bacteriophages with a binal symmetry. So this is another a complex one. Actually, this is quite iconic for a T cell fudge because they actually similar, in my opinion. They resemble a robot um, standing on spindly legs. But these long tail fibers are not used to walk. They are used to actually anchor themselves to the bacterial surface. And then you have the capsid head. So here is a viral enveloped by we have the protein spikes. Here is an example for this one would be the SARS-CoV virus. So many viruses are bound by that outer flexible membranous lipid layer. This is the envelope. So usually found in animal viruses because they emanate from the host cell plasma or organelle membranes from the host. So the viral envelope proteins are encoded, they are virally encoded. So they are the property basically quite a quote unquote copyright of the virus. Um, they are the spikes on the surface and it allows them to attach to the host cell. And that is one of the reasons why these viruses become infectious. And also one of the most major interest in developing vaccines, in developing antivirals or similar antivirals to these viruses, these surface proteins and these viruses because they allow them to infect specific cells. Now, let's look at the genomes of the viruses. The viruses contains their own genetic material, but they are quite diverse in the types of what exactly genetic. We do not say they have their own DNA because some viruses, instead of having DNA as the genetic material, they have RNA and they can be single or double stranded, plus sets or negative sets. So they are quite a variety. So the genomes are linear or circular, but some RNA viruses does not have a single molecule of DNA or RNA, but rather have segmented ones. And since there are quite a lot, not exactly a lot, but there are quite a number of viral proteins needed for the replication and, of course, assembly of the whole viral genome, or rather virion. The packing of these viral genes in their genetic material is very efficient. So some of them are overlapping, some of them are quite segmented inside their genome. So they are packed with efficiency in mind. Okay, now let's look at the viral life cycles. So the life cycle of a virus is quite let's say this is an interesting thing because we have um, a two-phase cycle. So, in the early studies on viruses, early experiments in 1939 by Delbruck and Ellis with a bacteriophage T4 E. coli. So, here is um, we have this duality in their life cycle. So, we have the eclipse phase and the burst phase. So, we have the eclipse where there is no less phage count, the virus acclimate or integrates inside, and then we have the burst period where the viruses kill off the cell and multiply rapidly and kill off the host. So this a one-step growth curve. So unlike you have infect, acclimate, then grow and kill the host type of life cycle. So this is the normal, or rather one of the first known type of life cycle for the viruses. So you attach the virus first attaches to the host cell. They enter the host cell. They hijack the cellular mechanism of the host cell to synthesize not the the proteins needed by the cell, but rather to synthesize their own, to synthesize other viruses. Then they assemble inside the host, and then virion release. So sometimes in the process of release, the cells burst. That's why it's called sometimes called the burst phase. Why does it burst? Because during the hijack phase, they synthesize many copies of themselves to the point that the cell just have to burst because they are too full. And now you have lots of virion particles to infect other cells. And then the cycle starts again. So let's look closely at this mechanism. So first is the attachment. So in order for a virus to become infectious, an effective infectious agent to the host, it must have um, some sort of attachment. It must be able to attach itself to the host. So usually through a receptor, a ligand receptor attachment. So the ligand from the virion will recognize a receptor from the host and be able to integrate itself inside. So that is why some viruses infect only, for example, certain species of plants and not others. It infects only animals and not plants, so on and so forth, because they are specific to their to that they specifically attach to the host through this receptor ligand receptor mechanisms. Now for these plants, because plant cells are covered by cell walls, plants become vulnerable to the viruses if the plant itself becomes damaged, or there is uh, the cell membrane, or rather the cell wall is damaged enough so that they can enter. So once they have entered into the host, they release their nucleic acid, their genetic material. So the entry is through endocyte fusion and endocytosis. We have a video added in your lecture folder for that, but basically it enters the cell through a cycle similar to an endocytosis. Then once inside, they release their nucleic acid. So here is the mechanism of this viral entry. Now, once inside, they synthesize their genetic material, and they also synthesize other particles, other viral particles to them. So basically, they synthesize all of the ingredients to make this virion. They replicate themselves. And then after that, they assemble themselves inside the cell. So once there are many of them inside the cell, to the point that the whole cell itself is very full to bursting, that the cell has no choice but to basically pop because they are too full of this genetic material. So that is one of the way, host cell lysis, or sometimes through budding. So the viral cells emanates from the cell membrane. They bud or arise from this cell membrane. So this is the budding. So they not they do not exactly pop the cell in order to be released. Unlike this one, the cell is popped in order to be released. But this one, they bud from the cell, they emanate from the cell. But in doing so, it kills also the cell. So this is the virion release. This is usually for envelope virus, where this the envelope is assembled in the integrated in the plasma membrane structure of the host. And so this is, and then they form buds. It's similar to an exocytotic behavior. So you have this formation of this bud and then onward to release. So let's look at the types of viral infections. So sometimes not all viruses cause an immediate death to the host. So we have a virulent phage, which are viruses that always kill their host. They have only one reproductive choice, and that is to invade and kill the multiply and kill the host. So they multiply immediately upon entering the bacterial host, and it bursts the host by lysis. We also have temperate phages, which are the most common types of viruses, reproductive viruses. So they have two reproductive options. First is to reproduce lytically, similar to a virulent phage. The second option is to remain within the host, remain dormant inside the host. And we call this phase as lysogenic phase. Lysogeny is the relationship between the temperate phage and the host, whereas the cell is infected, but it doesn't necessarily kill the cell. So the prophage, these viruses exist inside the host in the form of a prophage. And the host that is infected is a lysogenic. So for bacteria, it's the lysogenic bacteria. Here is an example of that is the HIV virus for the humans. HIV virus tend to lay dormant inside the host and only becomes virulent under certain stressful conditions. So here is a double life cycle, the lytic versus lysogenic. So both of them become infected, but in the lysogenic cycle, their DNA is incorporated inside the host. And since it's incorporated inside the host genome, their genomes, when the cell replicates, it replicates also the bacteria. So the rate of replication of this prophage virus is the same as the rate of replication of the cell. It's slower, but it doesn't kill the host. And it basically allows, basically, if you look at in an ecological perspective, we can say that viruses can be predatory because it kills uh, predatory parasites because they are parasites. They must hijack the host, but it also kills their host. But the more it kills its host, the less the population of the host will go down until there comes such a time that since all of the hosts have been consumed and therefore the virus itself becomes inactive or technically extinct, or it won't exist anymore because its host no longer exists because it has already eaten all of its or killed all of its hosts. And this is not quite effective in the larger scheme of things and in a longer time span if you consider it. That is why the lysogenic cycle, for a long-term perspective and in a larger view of things, it is a more efficient way to live for the virus. So they live together with their host cell in the form of their prophage. So this is your lysogenic cycle. So lysogenic conversion. So temperate phage changes phenotype. One of the downsides is to one of the downsides is the changes in the phenotype of the host, and sometimes they can be uh, actually it can most of the time it can be detrimental. But the lysogenic cycle, it allows them to remain inside the host, and they won't consume all of their host, but they still retain their high degree of multiplicity. And also, um, the lysogenic cycle is one of the things that makes viruses quite attractive to biotechnologists and bioengineers. Because if you want to edit out, if you want to incorporate external DNA from to a certain cell, we can use the mechanisms of the virus. Okay, so let's look at archaeal viruses. Archaeal viruses may be virulent or temperate, and they can also establish chronic infections. But again, because archaeals are difficult to study, their viruses are also difficult to study. Okay, so let's look now at the eukaryotic cells. So for the eukaryotic cells, the cytocidal infections results in cell death through lysis. So this is the virulent part of these viruses. But persistent infections in eukaryotic cells may last years because eukaryotic cells are multicellular, are part of a multicellular organism. Although it kills the host cell, it doesn't necessarily kill the organism. So there is that virulent stage here that can last years to in a virulent state with the host because again the host has many cells comprising it. So some cytopathic effects of this host can be degenerative changes or abnormalities, and some of them can also transform into malignant or cancerous cells. So how do we study this virus? So viral studies, we study them, we cultivate them in order to study them. We cultivate them in beds of cells because again, viruses are acellular. They are actually not living organisms. So they live inside their host. So we infect host cells and allow them to grow inside them. So the most common way, I'd say this is the most traditional way. We have an agar with a bed of cells and we infect them with the viruses. So these are plaques. These are regions where the viruses are, and cells do not grow because they have basically died due to the infection of these viruses. So we observe them in agar cultures. For animals, sometimes we also use fertilized eggs of chickens and infect them with viruses to observe cytopathic effects. For the plants, it's a plant tissue culture. So there are also some whole plant cultures of viruses inside the actual plants that provide localized necrotic lesions. So how do we quantify a virus? Through plaque assays. So you delete a viral samples, you cultivate them in a plaque assay and count how many plaques there are. And then we express them with plaque forming units because we suppose that one plaque equals one virion unit. So it's proportional to the number of the viruses from the original solution. So that's for the usual studies on the virus. So let's look at the other, aside from viruses, what other cellular entities out there. So we have viroids and satellites. So these are non-viral. They're not exactly viruses. They're smaller, much simpler than viruses, but they are genetic agents that can cause diseases or infections. So you have the viroids. They are simple infectious agents that consist only of RNA. They are closed circular RNAs, thus they are quite resilient to external forces because otherwise they would be easily destroyed. So they do not have any protein coat, and again, like viruses, they require a whole cell to replicate. They are usually seen in plants and cause plant diseases. And one of the ways to actually prevent them is to silence because there's RNA, so we can just silence them. And we have the satellites. So infectious nucleic acids like DNA or RNA, just like the viroids. So they all, however, they have nucleic acids. They are nucleic acids encoded in capsid and a helper virus to replicate. So they piggyback on a helper virus, but they encode their own capsid proteins. However, for the replication of the enzyme, unlike the viruses, because viruses inside their genetic material, they have their own um, enzymes for replications or even assembly. Some viruses, the satellites, do not have their own. So they need to piggyback on the viruses. So they are parasitic to the virus. But they do not infect the virus. They infect also the same host as the virus. But they require the virus to do that. And last but not the least are the prions. So aside from nucleic acids as infectious agents, we have the prions or the proteinaceous infectious particles. They are not nucleic acids that are causes infection, they are actually proteins. It's just a single protein. And the thing about prions is that they are quite difficult to decontaminate. In viruses, it's simple hand washing, washing with soap and water, or even alcohols and other cleaning solutions can easily destroy the virus particles. However, for these prions, not even heat can destroy a prion. Even cooking them does not destroy them. And that is actually why they are quite difficult to deal with. In order to destroy a prion, you need to have very high temperature, very high pressure, and an extremely acidic or an extremely basic environment to do that. Very, very strong uh, basic solutions to destroy them. They are quite tenacious. So examples of prions are the causative agents of scrapie in sheep, and you have the bovine spongiform encephalopathy or called the mad cow disease. And unfortunately, as a as a prion, as a protein particle, they can infect a wide variety of host organisms. They do not necessarily, prion proteins do not necessarily replicate. What happens with prions is that it's similar to having because they are made up of a protein that has a normal protein that has misfolded, that have an unusual or an abnormal folding conformation. When it is associated with other proteins, it destroys the original conformation, and the normal protein becomes a prion protein that is able to infect other proteins. So they convert other proteins to themselves, similar to like a zombie, for instance. So as basically you have one zombie, but it infects other humans. It becomes zombies that infects other humans. Basically something like that. So you have this prion um, uh, that self-propagates not by replicating themselves, but by influencing or converting other proteins to become like them. and so spreads the disease. And most of the causes of this disease, or one of the main targets, are the brain. So they have brain degenerative brain diseases and encephalopathy. So they produce holes in the brain, and of course kills the, well, quote unquote, the host. Although basically it's just a very abnormal protein that cause a very bad influence on other proteins. Okay, so those are the things, and these are the acellular entities that are in the microbial world, but not exactly part, not actually made up of cells. Okay, so that's it for our lectures for this module.